All right, let's move forward. And so I think we've got folks both in the room and also remote. Therefore, that's why I'm using the microphone. Welcome everybody back to the winter quarter and our first in-person seminar, which is going to be given by Anna Waldeck, who's now obviously a postdoc in our department. Anna did her undergraduate degree at the University of Chicago, right? So correct me if I'm wrong, because this is sort of on the fly. And then went to Harvard, to the Department of Earth and Planetary Sciences, to work with David Johnston. And so her work involves looking at all three of the oxygen isotopes, the stable oxygen isotopes in various sulfate minerals, and in part to try to reconstruct oxygen concentrations. And so I'm really excited to hear this work. I'm not sure we've heard triple oxygen isotope work here. Andy, have we heard this? Yeah, and so so this is exciting, no pressure. And so thanks everybody for coming, and thank you, Anna, for giving this, and we look forward to hearing it. Good luck.
Thank you.
This looks like an ornery crowd, so I don't know.
Okay. So I'll just start by saying thank you, Matt, for the introduction. Thank you to Maggie for organizing. And I would like to also thank everyone in my group from graduate school and the Johnston group and collaborators that have helped with various projects contained within this presentation. So I'll be presenting today my work from my PhD on the stable oxygen isotope composition in sulfate minerals and what these oxygen isotope compositions can tell us about Phanerozoic climate and atmosphere. So across the last 550 million years of the Phanerozoic, we have this predicted rise in atmospheric O2 levels. On the y-axis is atmospheric O2 concentrations in terms of present atmospheric levels. And these two curves reflect the COPS and geocarb geochemical-based models for predicting this rise in oxygen. And we think that it happened sometime between the earliest Phanerozoic and around 300 million years. And when exactly the timing and why, what drove this oxygen increase, is still a question that's being investigated in geoscience. So the important thing to remember about these models is, although they're based on geochemical records and our observations of the fossil record and rock record, these are not direct measurements of the atmosphere. So we only can measure directly the atmosphere in ice cores going back to around 1 million years ago. So today I will be presenting some new work on the triple oxygen isotope proxy, or CAP delta 17O proxy in sulfate minerals, which is thought to directly reflect the level of oxygen in the atmosphere. So this measurement, CAP 17O, when it's more negative, is thought to reflect times of low O2, and more positive is thought to correspond to times of higher atmospheric oxygen like today. CAP delta 17O is a relatively new isotope proxy. It's a measurement of deviation from a predicted relationship between small delta 17O and small delta 18O. So these small deltas are more commonly measured. And then recently, with improvements in instrument capabilities, we can distinguish deviations from this most common linear relationship that's found in nature. And so most Earth surface processes are going to have composition, oxygen isotope compositions that fall along this line that has a slope of theta or approximately 0.5. And whenever a measurement deviates from this relationship, it has a non-zero cap delta 17O composition. So for example, atmospheric O2 has a negative cap delta 17O. So when we look at the rock record, and these are measurements of sulfate minerals, and the cap delta 17O composition is on the y-axis. For a variety of sulfate minerals over time, especially in the Proterozoic, we find very negative cap delta 17O signatures. And so the interpretation that's made is that these negative cap delta deviations are directly reflecting atmospheric O2 incorporation. So I'm going to walk through now how this proxy is thought to work and the theory behind it. And so this happens through a couple of steps. So first, atmospheric O2 acquires this negative cap delta composition through stratospheric exchange reactions. So O2 in the stratosphere is constantly exchanging oxygen isotopes with CO2 through ozone and other intermediates. And this produces a negative cap delta 17O of O2. And the magnitude is related to this ratio of O2 to CO2 in the stratosphere. There's also, of course, in the global oxygen cycle, other processes that can produce oxygen, like primary production. And so this flux contributes oxygen with a cap delta 17O close to 0. So in interpreting the cap delta 17O composition of atmospheric O2, you need to consider both of these variables. So yeah, this is an example of how atmospheric O2 composition, the cap delta composition, is thought to vary across a range of O2 to CO2 ratios. And then what's not shown here is also that primary production fluxes can overprint the atmospheric O2 back towards 0 per mil. So yes, so based on our initial models, we have predictions for how O2 and CO2 levels have varied in the past. So here I'm just showing how the atmospheric O2 composition 250 million years ago or 500 million years ago is varying. So it's thought to be more negative in the past, and that's what has given rise to these very negative cap delta 17O compositions of sulfate minerals. So I'll move to the second step, which is how does this atmospheric effect become incorporated into sulfate? So it's thought that during weathering of reduced sulfur minerals, especially pyrite, that the product sulfate acquires oxygen atoms directly from O2 and water. And so this sulfate oxygen it's a mixture of oxygen from atmospheric O2 and from water. And the fraction of O2 incorporated into sulfate is like observed to vary across a range, but the commonly cited fraction is between 8 to 15%. So if you measure this sulfate cap 17O composition, you might be able to back out the cap 17O composition of starting O2. Yeah, so this riverine sulfate that's produced as a result of weathering is containing some fraction of this negative, like large negative effect from atmospheric O2. And then finally, after sulfates weathered through and carried by rivers to the ocean, it might undergo additional sulfur cycling processes that are not shown here before it's eventually buried in the rock record. So what we're looking at is these precipitated sulfate minerals, and we're trying to get information about this starting cap 17 of O2. So just to recap, we have this atmospheric effect generated in the stratosphere, and it's the magnitude of cap 17 of O2 is dependent on the ratio of CO2 in the atmosphere and primary production fluxes. Then we have oxidative weathering of reduced sulfur minerals, which can lead to O2 incorporation into sulfate. And actually, experiments have observed a range of fractions of O2 incorporation, anywhere between 0 and 60%. So this process is really under constrained at this moment. And then finally, these sulfates that are in the ocean and maybe undergoing additional sulfur cycling processes are eventually buried. And so what's interpreted is the most negative measured cap 17O value from any one time period is thought to most closely reflect this atmospheric O2 composition. And then more enriched cap 17O values are thought to be reset, and the consequences of those processes is less constrained. So really, when you look at this, what's being interpreted is just the most negative measurement from each time period. So the work that I'm going to present today is going to focus on this, mostly on this last step. So we are measuring marine sulfate archives and interpreting the cap 17O composition using our a context of the global sulfur cycle and the environment, what we know about the environment that this sulfate came from. So in my thesis work, we targeted 3 sulfate archives to understand how the sulfate cap 17O proxy works in the Phanerozoic. So we started with modern seawater sulfate, just dissolved in seawater. And then we moved to the marine barite record, which covers the last 150 to 200 million years. And then finally, we zoomed out to the entire Phanerozoic by looking at the marine evaporite record, which is it's a more sporadic record. So you're not getting great temporal resolution, but you can go way further back in time. So starting with modern marine sulfate, because we want to root this proxy in the modern system. We have a prediction from our theory that this sulfate that's being weathered and by rivers and brought to the ocean is going to contain a mixture of oxygen from water isotopes. So this is the global meteoric water line in terms of cap delta 17O and little delta 18O. And sulfate, that's the rest of the sulfate should incorporate some atmospheric O2 contribution. So this is our prediction for sulfate that contains anywhere between 10 and 25% of its oxygen from atmospheric O2. So we went and measured marine sulfate in 2 ocean time series sites, and this is the cap 17O composition and little delta 18O composition. And you can see that it's consistent between sites. It's also not really varying much across depths. So we interpret the modern ocean as well mixed, which makes sense. Sulfate is very rich in the modern ocean, and the residence time of sulfate is much longer than ocean mixing. So then we go back to our end member plot our modern marine sulfate measurement plots over here. So it's not really falling between these end members as we would expect. So we have some inconsistency with our theory, which the theory predicts the sulfate in the ocean is going to be slightly negative, and our measurement was slightly positive in cap delta 17O. So we zoomed out again to the global sulfur cycle. So we have this sulfur weathering flux, and then once sulfate in the ocean is being microbially recycled, and this flux is, the estimates on this flux vary by orders of magnitude, but it's thought that it's a very large turnover of sulfate in modern. So this is 8 teramoles per year, as opposed to around 3 teramoles of sulfur are weathered every year in the modern. So we wanted to put isotope constraints on this microbial flux. And we can do this because microbial sulfate reduction has been so well studied for many decades. And what's known about this process is that sulfate is reduced sequentially to hydrogen sulfide mainly in pore waters. And this happens through a series of sulfur intermediate phases. And these intermediates, especially phases like sulfite, through which most of the sulfur is going to pass, is exchanging with water. So we have a prediction based on measurements of pore water and theoretical measurements of isotope equilibria for what this sulfate microbially cycled sulfate flux oxygen isotope composition should be. So we think that it should be close to 25 per mil. And we have a cap 17O prediction as well that's slightly negative. And it will vary depending on your water composition and your temperature and a couple of other environmental constraints. But we know that the delta 18O is going to be enriched, and cap 17O is going to be slightly more negative than seawater sulfate. So when we plot a theoretical prediction for this sulfate equilibrium on our 3 isotope plot, you can see where it's falling. So it's enriched in delta 18O and depleted in cap delta 17O from modern marine sulfate. So we're still not exactly able to explain the modern marine sulfate composition using these end members. We really are looking for something with a positive cap 17O composition that's influencing marine sulfate. So recent work from the Johnston lab has measured a riverine sulfate data set, all these yellow triangles. And this is sulfate from a pyrite weathering dominated river system in the Himalaya. And all of this river sulfate is enriched in cap delta 17O. So this is in contradiction to the theory that we had started with, where we were incorporating very negative O2 into sulfate. And so it's falling above our modern marine sulfate composition. And we thought this was very interesting because you could explain all of this data using a linear relationship that encompasses rivers and modern marine sulfate. And so the way that we ended up interpreting the modern marine sulfate is that it's not actually incorporating much, if any, resolvable atmospheric O2 signal. So we instead interpreted it as a balance of the 2 largest sulfur cycle fluxes, the riverine flux, which is constrained with this data set, and microbial flux. So then we wanted to move back in time because we're still trying to get at this question of like, why in the Proterozoic can we measure these very negative cap delta 17O compositions? So we started with the marine barite record. Marine barites are a high temporal resolution, very robust to diagenesis record, and they are constantly forming in the open ocean water column when respiration creates this barium-rich fluid, which mixes with sulfate-rich ocean, and you form these barites that are very resistant, and you can find them in ocean sediments. So we have a nice time record of the last 150 or so million years. So we went and looked at the barite record from many different cores around the ocean, representing different parts of the ocean that were consistent with each other. And we have this prediction for how the delta 18O composition of marine sulfate has varied through time over the last 130 million years. And so we also measured the cap delta 7O composition, which I'm not showing yet, but our expectation going in was to try to see if some atmospheric O2 was being incorporated. And so this is our prediction based on how O2 and CO2 have varied over the last 130 million years for what we would expect marine sulfate to look like if it was just incorporating like a constant 10% of this O2. So the record that we measured is sitting above this O2 incorporation line, and we don't really interpret the shape as reflecting this O2 trend through time. So we think that's similar to the last 130 million years of marine sulfate are not incorporating this atmospheric anomaly. So going back to our triple isotope plot where we have cap delta 17O versus small delta 18O, we wanted to see where the barites fall, and they fall to the right of this riverine end member. So the way that we are interpreting this right now is that we have this positive riverine end member, and then marine sulfate is being overprinted towards a sulfate water equilibrium isotope end member. And then this shared process or framework has been consistent over the last 130 million years. So really, instead of a story of atmospheric O2, the cap 17O record is telling us something about the proportional influence of weathering and biological sulfur cycling over this time interval. So we modeled based on the oxygen isotopes and assuming oxygen isotope compositions of our fluxes, we can get a prediction for this relative influence of biology and riverine sulfate. And this is over the last 100 million years. And so we see a pretty steady ratio. We are interpreting this time in the Cretaceous of very high biological sulfate turnover relative to riverine sulfate weathering. And then this slight decreasing trend represents increasing relative influence of riverine sulfate over the last 40 or 50 million years, which would be consistent with other records showing an increasing weathering flux of pyrite as the uplift of the Himalayas over the last 40 to 50 million years. So next, we took as an input this constraint on JRIV, the riverine sulfate weathering flux over time. And with this independent constraint, we can then form a prediction for this microbial sulfate flux through time. So we're thinking about this now and what the implications would be for the relationship between sulfur cycling and sea level changes and shelf area changes. So just to really drive it home, marine sulfate over this time interval, like we're not seeing significant atmospheric incorporation, this predicted negative cap 17O. Instead, we're interpreting other processes in the sulfur cycle. So we still have this question of how the heck do these Proterozoic sulfate minerals record such negative cap 17O anomalies? So the marine sulfate and marine barite record in younger ages is sitting up here for context around 0. And so what we did next was look at marine evaporite minerals. So these marine evaporites extend further back in time than our ocean sediments extend. And they're measured also throughout the Proterozoic. So we want to interpret what kind of environmental information are they recording. So what we know about evaporites is that they form in these restricted ocean basins when there's a lot of high temperatures, evaporation, leading to very saline restricted basins and precipitation of ocean salts. So the thinking was that this sulfate should pretty closely reflect seawater sulfate. So we started out with a more modern, more recent 6 million year old evaporite basin to test whether marine evaporite cap 17O reflects seawater sulfate at the time of its precipitation. So this is work that I did with with a collaborator, Haley Olson, another grad student in the Johnson lab. And we sampled 3 subbasins from around the Mediterranean Messinian basin to see whether these 3 subbasins were recording a consistent sulfate signature and whether this sulfate was similar to the ocean at 6 million years ago. So we have an independent constraint on marine sulfate composition from similarly aged marine barite minerals over this time period. So the delta 18 is we think of Messinian aged marine sulfate is around here. And the cap delta 17O is a little bit enriched over 0. The compiled evaporite measurements that we have are overall enriched in delta 18O and slightly depleted in cap delta 17O from the contemporaneous seawater. And this delta 18O is an effect that has been observed before or like hypothesized before. So when we look at these same measurements in triple oxygen isotope space, so we have the cap delta 17O composition on the y-axis and small delta 18O on the x-axis again, you can see the direction of offset. So starting marine sulfate is up here, and then these basins are falling to the lower right, and they're on the trajectory towards this predicted equilibrated sulfate end member. And so we're interpreting these measurements as reflecting a mixture of marine sulfate and sulfate that's been overprinted. So during the time that this basin became restricted, and is evaporating. We believe that there's some microbial sulfur cycling probably going on in the basin that's overprinting the sulfate towards more negative values. So this was interesting to us too, because not that it would be an apples to apples comparison, but in the Proterozoic, you would always interpret the most negative value as most closely reflecting the atmosphere. But here we're saying these microbial flux is pushing us towards more negative Cap 17O compositions. Yeah, so just to recap, we have measured and constrained this offset of evaporate sulfate from marine sulfate. It's a pretty sizable offset considering how much sulfate we think is really, what we know is really in these evaporate basins, just these massive sulfate deposits. So this is like a very significant amount of microbial sulfur cycling. We're shifting the isotopic composition towards this equilibrated end member. And now we're sort of revising the framework for our interpretations of marine evaporate. And we're interpreting here the averaged basin value for Cap 17O. Because I think I forgot to mention, but we're interpreting these basins as well-mixed sulfate reservoirs. So we see sort of like normal distributions and not a lot of variation through stratigraphy. So we expect that these are very well mixed, just massive sulfate reservoirs. So zooming out to the Phanerozoic, we wanted to see if we could fill in some more information using the Phanerozoic evaporate record and try to understand whether this proxy is giving us information about atmospheres on longer timescales. So we're starting with the barite record of marine sulfate is sitting up here in the purple. And these circles are the same Proterozoic evaporate record I showed before. Now we're just taking the average, and this line is a measure of the distribution for each basin instead of plotting each individual point. So you can see that like there could be a slightly increasing trend over time. We added data from 9 new evaporate basins across the Phanerozoic and latest Ediacaran. And now with this added information, it makes it even more clear that there was a significant shift in marine sulfate Cap 17O sometime between this basin and this basin, or 320 and around 430 million years ago. And so we are interested to see whether this shift matches our predictions based on atmospheric oxygen, the atmospheric oxygen predictions over the Phanerozoic. So, oh yeah, and also we looked at how these evaporates look in triple oxygen isotope space. So this is back to our triple oxygen plot. So all of the modern data and marine barite record are all consistently following along this roughly linear trend, which reflects a shared process. And we think it's reflecting this overprinting from biology. And interestingly, that the averaged values of oxygen isotope composition of each of these basins in the mid to late Phanerozoic is falling roughly along the same slope after before around 430 million years, we do observe the basins to start falling below this line. And we can interpret this as potentially shifting towards the atmospheric O2 end member, which would be down here. Yeah, so we're going to test the timing of the rise in atmospheric O2. So based on the COPES model, it's thought to occur sometime around this 450 million years. And so using their predictions for O2 and CO2 and GPP through this, or primary production through this time, we can get a prediction for what CAP 17O composition of atmospheric O2 might have been through the Phanerozoic. And then we scale this to our evaporate record. So this is not representing the full magnitude of O2 change, just scaled to sort of be on the same frame as the evaporates. And we do see the shift around a similar time. You could say this point might be inconsistent with the record, but instead of focusing on the magnitude of change, we're more interested in the shape and timing. So it would be really interesting next to focus more on this interval of Phanerozoic evaporites, getting some more detailed work on basins in that time period could really nail down exactly when this transition occurred. And if it's linked to O2, it can be connected to the rise proliferation of land plants on the continents, which would have added an additional O2 flux to the atmosphere and also overprinted the CAP 17O composition of O2. So yeah, this was sort of the point that we're at now with interpreting this evaporate record. So just to wrap up, we started out by wanting to understand whether marine sulfate minerals are reflecting information about atmospheric O2 compositions. We started with modern marine sulfate and sort of worked our way back across the last 300 million years and found that all of these marine sulfate archives are similarly not incorporating resolvable amounts of O2. And instead, we can interpret their shared linear dependence in triple oxygen isotope space as a balance of influences from riverine sulfate weathering and microbial sulfur recycling. And then finally, in the early Phanerozoic, before around 430 million years ago, we are interpreting a shift away from this linear mass-dependent framework. And so we are starting to interpret this as potentially reflecting some O2 incorporation, and this could correspond to like the timing of land plant proliferation in the Phanerozoic. So I think that really getting a more, I did present a lot of caveats about this proxy, but I think that the relationship we observe is very powerful. Like these average CAP 17O compositions from many evaporate basins. Instead of just interpreting one outlier, we're actually seeing in an average of all of these tens of measurements from each basin, we see a real shift in CAP 17O compositions. And it will be really interesting to see if we can get some more detailed work from this time period. And I think that coupling this proxy with other geochemical redox proxies will provide a more complete picture of global redox budgets and atmospheric compositions. So yeah, that's all that I have. Little fast on time, but thank you.